about 1 hour, and then Dr. Sukla will come, and I think he is supposed to give lecture on metagenomics tools, something. So he will do later. So I can at least discuss while others are coming regarding fold change that I already discussed with you. So can any of you explain what is fold change? Fold change in RNA expression. So what does fold change mean? What it means? Yeah, so what is the meaning of upregulation?
So when there is a showing expression more than the normal expression.
Okay, so more than, okay. So when you say a gene is expressing more relative to control, so what it means in terms of RNA transcript abundance? So will there be more RNA transcript abundance or less RNA transcript abundance? More, right? So you would want to elaborate further that fold change is a ratio of normalized counts from condition and normalized counts from case, okay, or control. Sorry, normalized count from case or condition and normalized count of control. Okay, so yeah, case is condition actually. So okay, good. So clear? Yes. So and before we compute fold change, why we would want to obtain the normalized counts? There.
Multiple reads in the same transcript.
No. Why you would want to perform normalization of RNA reads frequency? No. Yeah, that you already do. Why do you want to perform normalization? Go back.
Some genes have 10 times sequence, some genes have 1 time early sequence. So to make the sequencing data.
Okay, yeah. So yeah, not gene actually, sample sequencing. You have to say because samples are sequenced. So bulk RNA-seq. So entire sample is sequenced. So some samples may be sequenced more, many-fold times more than others. Okay. For example, you have a sequence and that generated 100 millions of reads. Okay, and the other sample might have been sequenced 10 times more of that. So because of the depth of sequencing, there would be a difference in the counts. And so that difference is not because of difference in expression of genes, it is because in the difference of depth of sequencing. Clear? So make sure that, yeah, you want to explain in right ways. So it is clear that, okay, so I think, yeah, you did okay actually, but yeah, you have to say that samples are sequenced, so and there could be a difference in depth of sequencing. So one region, the other region could be the size of the transcript. So longer transcripts will generate more reads and shorter transcripts will generate less reads. So that difference is arising because of the difference in length of the transcripts, not because of the difference in gene expression, right? So you would want to control these two, so that what you observe, that is the difference, is actually due to the expression of the genes. Okay, so you would want to control it. So that is normalization, and there are several metrics that quantify this normalization, that removes this bias, and those metrics are RPKM, FPKM, TPM. Okay, so these are doing the normalization with respect to the length of the transcript as well as the sequencing depth. Okay. Okay. So basically, then when you have the normalized counts and you want to compute the fold change, then in the numerator you have the normalized count for the condition that you are interested in. For example, as I mentioned, that there is some disease that has happened to some individual. So you would want to, so for some gene, fold change. So normalized count, and I would say, let's say normalized count FPKM or RPKM. Okay, so FPKM of condition, and maybe let's say condition is some disease. So disease condition. Okay, and divided by FPKM of control. Okay, and control is healthy. For this example. Okay, so this is fold change. So basically, you are taking the ratio of the normalized count of the condition or case and the normalized count of the control. In this example, condition is disease condition and control is healthy. Okay, so you compute this, and for example, for a gene, this could be 1000 and this is 100. So what is the fold change? 10. Okay, so you will say that the this gene is upregulated 10-fold more. Okay, or the number of transcripts from this gene is 10-fold more for disease compared to control, compared to healthy. Okay, you can say that. Or sometimes people just say 10-fold more upregulated. On the other hand, for some other gene, it could be, I will just reverse it. It could be for some other gene. In the condition, disease condition, it is 100. In the control, it is 1000. Okay. So what we will say? Yeah, 10-fold down regulation. So the number of transcripts is 10-fold less in disease condition compared to the healthy. Okay, so down regulated. Both could be responsible for the disease phenotype. Okay, it is not just that the upregulated genes they are underlying, they are responsible for the disease. Those that are upregulated and downregulated, both could be responsible. Okay, so you and that is why you say differentially expressed genes. Differentially expressed genes means both types of genes, upregulated genes and downregulated genes. Okay. Okay. So and if you take a log of this, logarithm of this, then log fold change. Okay, and this is log, and what would be this? -1. Okay, if this is base 10, right? -1. Okay, so typically it is base 2. Most of the time people use base 2. So you have a metric that is called logarithm of the fold chain. You got a value, and of course you can say that a gene is upregulated by X fold, or a gene is downregulated by X fold relative to the control. But you would just wonder whether this is significant or not. Okay, whether this value happened just by random chance. So for that, what you do? I'm sorry. Calculate p value, right? So how you calculate p value? So, okay, so simple things. So you might have done a statistical significance test. Have you heard about a standard distribution or normal distribution? Okay, so so something. Okay, I don't know if I can make so. Yeah. Okay. Okay. And so let's do like this. Okay. And let's say that the metric follows this distribution. And and so you want to find whether a value is significant or not, right? So up regulation would be towards right or towards left, right? So high value, right? So okay, so you will compute p value for that, and p value, so p value is basically this area. Got it? You said p value. So p value is this area. For down regulation, where? Left or right? Left. So here actually. So here, and let's say that value is this. Okay. So this area is the p value, and you would have set a significance threshold or significance level, right? So for example, 0.01. 10%, no, not 10%. So yeah, 1%. Sorry, yeah, 1%. So 0.01. So and it is two-tailed test. So what you would want to do? What would be the threshold here? So 0.01 you will divide by 2, right? So 0.005. So let's say that is this, okay. So which means that area to the right of that would be 0.005, okay. So and here is also two-tailed test. So 0.005, and then you ask whether this area, which is the p value you got, whether this is less than significance threshold or not, okay. This is how you do the significance test, right? So if it is less, then you conclude that the value is statistically significant. Clear? So this is how actually you do test. Your null hypothesis is that the two genes have similar gene expression, okay, or equivalent transcript abundance. This is the null hypothesis. And then you test and you reject the null hypothesis. If the null hypothesis is rejected, then the fold change or whatever is the metric that you are using will be will be significant. So null hypothesis rejected, which means that it's significant, right? Because null hypothesis is that both are equal. Transcript abundances are equal. Okay. So if reject, it means that not equal, insignificant. Okay. So and the distribution actually is negative binomial distribution. It is not normal. So the distribution for logarithm of the fold chain follows negative binomial distribution. Okay, it has been established. So that is being used for assessing the statistical significance of a value of log fold change. Clear? So this is how it is estimated. Okay, so yeah, Dr. Sukla asked me to explain this. So I thought that, okay, you all understand. So he said that no, maybe some understand, some may not. So I thought to explain to you. Okay. Okay, so let's keep going actually, and because I want you to do some tutorials also, some questions that we will discuss. And yeah, this is the pipeline that my PhD student has set up for you using RNA-seq data from a bacterium. And he told me that it's a small data set, so students should be able to download and run. But still it is so large actually. I tried to do myself, and Galaxy was giving some error. So I talked to him and he said that don't worry, let students know that they may get server error, but if they keep repeating this 3 or 4 times at different times, then they should be able to run this pipeline, okay? Because he has run it and he has got the results, so it should run. So what you will do is you will input these accession numbers into this box. So 2 accession numbers are for the control wild type of this bacterial strain, and the other 2, so do not do more than 2, because this is only for learning. Once you have learned, you can then do for as many replicates as you want. So I am considering only 2 replicates. He did for 3. So I told him that it is possible that the pipeline will accept 2 replicates. Okay. So the more the replicates are, the more confidence, statistical confidence you have in the results, right? Statistics is better when you have more data. So, so what you will do is you will input each of these accession numbers. 2 are for control or wild type, and the next 2 are for this strain treated with hydrogen peroxide H2O2, which means that these strains have been subjected to oxidative stress. Okay, and then you would want to know which genes are upregulated and downregulated, which means differentially expressed during oxidative stress. So this is the problem that you would want to solve. So for each of these accession numbers, you will search for the FASTQ files. Okay. So copy paste the number here and then click search, and then you will get the sequences, nucleotide sequences. Select the first one. Okay, click on the first one, and it will take you to the FASTQ files. Can you see the FASTQ files? Okay, so there is just one FASTQ file. So you will click on this. This will download. Okay, so don't do now. This is for homework. Take home, yeah, because it will take a lot of time, and these are very large files. So just wanted to show you. And then once you have downloaded, so you will download for each of these accessions. So you will download 4 FASTQ files, 2 for control and 2 for stress, okay, condition. Okay, so and actually I have done some changes. So I'm going to send this file to Shravani, and she will forward to you all. Okay. So and then you upload these files here. So click here on upload and upload all these 4 FASTQ files. Okay. Okay, so then it will show here. So, so this is what Ahmad did. Actually, he uploaded 3 control files and 3 stress files, 3 condition files. Okay, and he renamed. You can also rename so that you are able to recognize which are control and which are conditions. Okay, condition ones. Okay, so 6 files are here being uploaded. 3 are control files and 3 are condition files. And then click start, then it will upload all the files. Okay, so these are uploaded. So it will show here. This is Galaxy that we were trying. So Galaxy platform. Okay, so Then these files are now uploaded. So I think it is better here for me. So the next thing that you will do is use a Trimomatic tool, and you can find it from all tools. Trimomatic tool is used for trimming the reads, because at the ends the bases have typically low quality. Okay, you would want to trim them. So Trimomatic is used for trimming the low quality bases as well as adapters. It can remove the adapters. Okay. So you will select Trimomatic, and then because these data were single end RNA sequencing data, so you will select single end, and then you will add all these files, 3 control and 3 condition files. Okay, and then run the tool. And these are default setting, you can let it as be. So when you will run the tool, then it will do the trimming and it will output the trimmed files. Okay, after subjecting to trimming of the poor bases, poor quality bases, and the adapters. Adapters are removed. Okay, so once you have the trimmed files, you would want to perform quality check. And for quality check, so next actually, for quality check, you would want to consider the fast QC tool. Okay, so select fast QC tool, and then add here actually in this box all the trimmed files. Okay. So these are all Trimomatic files. So add them here and then run. So when you will run, then it will generate the fast QC report. Okay. So for example, if you look at the report of one of the files, then if you observe like this, they are in the green zone, which means that it's a good quality sample. Okay. So good quality sample. So it is important that you have throughout good quality samples, otherwise your results would be influenced by bad quality bases. So it is important for you to assess this. So once fast QC is accomplished, then the next is because remember, you have to, so these FASTQ files have reads. These reads have to be mapped onto the genome, right? So you would want to download from NCBI, you would want to download the genome of this strain and genome as well as annotation, because you want to know about the genes. You want to quantify the expression of the genes, right? So you would also want to get the information about the genes. So that is called annotation. So you would download both the genome and annotation file. So I think this is a page now you are very much familiar with, right? The genome from NCBI. So click on download and download only. So click on download and download only genome file, FASTA file, and annotation file, which is GTF file. FASTA and GTF. Okay. So one is genome and the other is annotation. And annotation means features, the coordinates of the genes, because you want to quantify gene expression. So you have to provide this information to this pipeline where the genes are in this genome. Okay. Okay. So next you have to upload the genome and GTF file. So you will click on upload and you will upload genome and GTF file. GTF is annotation file. Okay. So which is shown here. Yeah, I think the resolution is a bit bad, but I think it's okay. So once you have done it, you would want to align the reads against the reference genome. You have uploaded the genome, you want to align the reads which are in FASTQ files onto the genome for each of the samples, for each of the replicates. Okay, for that you would select the tool HiSet2. Okay, this is for alignment. So select HiSat2, and then use a genome from history. So when you will click on this, you will see the name of the genome that you have uploaded. Okay. Okay. And then you would add here all the FASTQ files. So these are trimmed files. You will add all those files. Okay. These are the read files. And then run the tool. So when you will run, then the reads will be aligned against the genome, and BAM files will be created for each of the samples. Okay, BAM files are read alignment files. So read aligned, and then you can quantify how many reads have aligned to a gene or transcript. Okay, so first you have to select HiSat tool and perform read alignment, which will generate the BAM files or the alignment files. Once the alignment files have been generated, then there is a tool called feature count tool. So you want to compute the normalized counts, right? RPKM, FPKM, or TPM. So yeah, this is a tool for generating the counts for each of the transcripts. So you will select all BAM files, all files generated by HiSat tool. You have selected feature count tool, and then you will just run the tool. It will generate the feature counts for each of the samples. Okay. Okay. So and one more thing that you will do is you will upload the GTF file, because unless you upload this, how the program, how the pipeline will know about the genes, right? So annotation file you have to upload so that the program knows that there are n number of genes and these are at these positions in the genome, and it will quantify then the count of reads mapping onto each of the genes. Okay. Okay, then run the tool. When you will run the tool, then okay, fast. When you will run the tool, then okay, here it is. Okay, then you will see the count for normalized count for each of the genes. So these are gene IDs. So this gene has 137 counts. Okay, count of the reads aligning onto the gene. Okay, so this gene has 130 reads aligning onto it. The next one 66, 38, and so on. Okay, so these are the reads that align to each of the genes. So once you have this, then you can perform differential expression analysis. So in the differential expression analysis, as I was explaining on the board, you compare the normalized counts of condition with case, with control. Okay, normalized counts of condition or case with normalized count of control. So that is fold change. Okay. So that will be done by this tool called DE-seq tool. So look for a DE-seq tool in this all tools tab, and then select this, and then write a factor name per your project data. So this is actually Bacillus anthracis. So you will mention that here in this box, and then you will select in the first factor level all the control count. So there are 3 control samples, and you have got the counts for them, right? Until now you have the counts. So you will add those count files. So this is for control. So 3 control count files added. Okay, and then next you will add the count files for the condition. Okay, first control. So the strain was not subjected to stress, control, and the next one is subjected with the hydrogen peroxide stress. Okay, that is the condition. So you will add all the three count files from the condition, okay? Because you want to compare these two. You want to compare the condition with the control. So first you add a control, and then you add, just below it you add the condition, the stress condition files, okay? And these are normalized count files generated by the program, and then you will run the tool. Tool should produce the log fold change, p value, and so on, okay? So run the tool, click on run tool, and then okay, and you have the option to select different output files. This is generate plot, so I can't read this, okay? And, don't worry actually. So run the tool, okay? Run the tool. I can read there but not here. So you have different options for output. So you can get output for normalized count file. So this file will have normalized counts for each of the genes, for control and for condition, okay? And the other output file is log normalized. So take the logarithm of the normalized values. That would be log normalized. So you will get another file, and then this is the output plot file. So DC2 was able to produce the PCA plot, principal component analysis plot, based on the normalized counts, okay? And you can see that the control samples, they group together, okay, as you would expect. Control samples group together, and the condition samples, the stress condition samples, they also group together. But still actually these two are grouping well, but this is a bit far. But you would expect that the control is separate from the condition, which is reflected here, okay? Okay, so and then you can also generate volcano plot. So for that you would want to input the normalized count table from DSIC2. So we have already the normalized count file generated by DSIC2. So you will add it here and then run tool, okay? So when you will run the tool, then you will have the options. So whatever settings you want to have, you can change here. Otherwise, continue with the default. So here is the volcano plot, okay? So on x-axis is log fold change, on y-axis is minus logarithm, actually this is better, minus logarithm of the p-value, okay? So if p-value is small, then minus logarithm of p-value would be, so if p-value is very, very small, 10 power say 100, so logarithm of that would be base 10. Logarithm of that would be, so okay, so log p value, and that p value is 10 power minus 100, okay? This is what is there actually, minus log p value. So this would be minus multiplied by 100, right? And this is 100, okay? So which means that, and if this is 1000, then this would be 1000, right? So as p value decreases, this is increasing, right? So the p value is 10 power minus 1000, then this value is 1000, okay? So which means that the lower it is, this metric is going up, right? Going higher. So just to help you understand volcano plot, So then you would want to look for here itself. You would want to look for the genes that have fold change indicating up regulation or down regulation, right? So relative to the control. So which would be, so if it is neither up regulation or down regulation, what would be the value? 0, right? So it's a ratio. So 1 and log 1 is 0. So it should be significantly higher than 0 or lower than 0, right? The log fold change. So towards the ends of this x-axis, and the logarithm of p value should be higher. Okay. So basically, then you are looking in this region, and this region is down regulation or up regulation, down regulation, and you are looking in this region. So the two extremes on this axis. So as extreme you are, the more confidence you have in the significance of the gene with respect to their differential expression. Okay. Okay, so and they are indicated by red. So red ones are up regulating and blue ones are down regulating. And you can get a list of these genes, differentially expressed genes that are up regulating and that are down regulating. So you will get a file, actually that would be tab delimited, I think Excel file, that will give you the fold change, the log fold change, p value, and so on. Okay, all this information. So you will know that which genes are up regulating and which are down regulating under the stress condition. Okay, and of course you can generate volcano plot to show which genes are up regulated and which are down regulated. Okay. Okay, so and you can also generate heat map. So for heat map, you will click on heat map 2, and then you can add the normalized count data. So logarithm of the normalized count data, and then just run it. So when you will run it, you will see the clustering map, the heat map. So the genes with similar expression, they are clustered. So it is a tree actually. You can see the clustering as a tree. Okay. So those genes that have a similar expression, they will group together. They will form a clade. So which is indicated here. So here is the tree for genes with their normalized counts in different samples. Okay, 3 control samples and 3 condition samples. And at the top, you have the samples that have also been grouped. So in the heat map, you can do grouping for both the samples as well as the genes. On the top, you have the grouping for samples. So you would expect that the controls should group together. Okay, and they did actually. You see the tree, and the condition samples should group together. They did. Okay, so as expected. But more often you would focus on the grouping of the genes. You would want to identify the groups with groups with genes with similar expression pattern, because often you would want to know that which pathways are regulated during stress condition. You would want to identify those genes involved in certain pathway, right? And they may be there in one of these groups. So you would like to know what these groups are. You would want to perform functional and enrichment analysis of these groups to check which functions are enriched in these groups, which pathways are enriched, so that will help you in understanding not just which genes are differentially expressing, but which pathways are differentially expressing, right? Okay, so yeah.
So if we want to narrow down and check some 5 or 10 genes, then how will you?
Oh, you want to narrow down to genes. Oh, okay. Sometime what people do actually, they just do it based on a fold change. So they want to have the genes that have the highest fold change. Both way actually, highest, upregulated and downregulated. Okay. But often the genes that may have moderate fold change or even lower than moderate, but still significant, they may be playing major role. So what we have done actually is that we developed a new method published last year. So after you have identified the genes that are differentially expressing, for example, this bacterial strain. So even before doing that, we make a gene network, which I think Sravani you are working on. So we make a gene network from RNA-seq data of this species representing many different conditions. Okay. So in the gene network, it is just like a graph. Two genes that have similar expression, they will get connected. Okay. Based on correlation of the gene expression. So if it is say above certain threshold, you will connect those genes. Okay. So you will make a gene network, and then you will decompose the network into groups of genes or modules of genes, and those modules could be representing pathways of genes or single pathway or many pathways that are cross-talking. Okay, they may be represented in those modules. So, and because genes in those modules are connected under many different conditions, which means that these genes are regulated under so many different conditions. So, it's not specific to one stress. Okay. So once you have the gene network, then you can start mapping the differentially expressing genes that you have obtained from your experiment for this particular strain. Okay, you will start mapping onto this network, and then identify the modules that have an enrichment of differentially expressing genes. Okay. So you identify the modules in the network with enrichment of differentially expressing genes, which would indicate that those modules are differentially expressing under this stress condition. Okay. So sometimes you would want to go above genes, because when you do RNA-seq experiments, then you can have thousands of genes differentially expressing. For example, if you are doing for some plant species, there could be 10,000 genes that are differentially expressing, and it's a challenge to interpret this. But when you will make network and you will map the differentially expressing genes on the network and identify the modules that are enriched in differentially expressing genes, maybe you have found only 4 modules that have enrichment of differentially expressing genes, and they are representing these pathways. So you would want to perform the gene ontology or functional enrichment analysis of those modules to understand which pathways or functions they represent. Okay. So that way you will come to know that these pathways or these functional modules, these are differentially expressing under that condition. So now you are looking above gene, and that's important, because often genes do not work alone. They work together, they interact and confer a product or certain phenotype or some signal, right? So this way you can narrow down. You can narrow down, and the way people narrow down, I think that's a very naive way of doing it. That I am going to take top 10 upregulated genes. I am going to take top 10 downregulated genes. That's not the right way. Okay, because it is likely that the major phenotype being observed that is not arising from them. It could be arising from some moderately expressed genes. Okay. So that is why it is important that you go beyond single gene analysis. You start doing pathway or functional analysis. So above the gene. Once you have found the genes, then you have to find the interactions between genes, identify the pathways that are differentially expressing under certain condition. Okay. So yeah, if you are interested, then you can read my paper published, I think, in 2023, a gene network for Pseudomonas aeruginosa. Go to PubMed and you will find it. So I can show you that. PubMed. PubMed, and then okay, and I will do most recent, and just scroll down. I think it was published last year, 2024, 2023. Disease net. Oh, okay. Oh no. Yes, here it is. Okay, so yeah, you want to take a look at this paper published in M Systems, a gene network driven approach to infer novel pathogenicity associated genes, application to Pseudomonas aeruginosa PA1. So this is a very well studied pathogen, Pseudomonas aeruginosa PA1, and virulence genes or pathogenicity associated genes have been exhaustively identified and studied. But we saw through this approach that novel genes can be discovered, and not just genes, but their network can also be discovered using this network based approach. Okay. So you'd want to take a look at this paper if you are interested.
So does this work for more complex organisms also or only for bacteria?
No, this is a generic method. It can work for any organisms. You only need to have tons of RNA-seq data for that organism. Okay. So then you can use this pipeline to identify novel genes involved in certain process, and also the pathways or gene networks that are differentially expressing under certain condition. Okay. Yes.
If we have RNA-seq data, that
Yeah.
is not differentially expressed, just RNA-seq.
Right.
And if we want to track that which transcripts from the whole transcriptome are coding for, like, are follow particular metabolic pathway,
Yeah.
how can we identify those transcripts from the transcriptome?
So certain specific metabolic that you know.
Yeah.
Oh, so I think that if you know that, then you know the transcripts, right, that are associated. So you can also look in the list of differentially expressing genes whether those transcripts are there or not. Okay. And when you will make the network, and if those transcripts map onto certain modules, then you know the interactions of those transcripts with other transcripts, right? Because the modules represent the interactions.
I don't know which transcripts are those. I want to find out the transcripts.
Oh, you don't? Yeah, you don't. Yeah, you don't know actually. Yeah, that is fine. But you know the functions, right? Okay. So you can then screen for functions. Functional analysis you can do, and so there are programs that can annotate functionally, and then you can do the functional enrichment analysis or pathway enrichment analysis of the modules, and see if those functions are represented in those modules which have enrichment of differentially expressing genes. Okay, so this way you may be able to identify. So, so there is.
There is integrity pathway analysis.
Yeah, yeah.
It's very costly for a student.
Yeah, very. Yeah, right. And so that is from SRA International, right? Yeah, I think that is subscription. And I also try to the, for example, flux balance analysis. And they ask for money. So I tried, I couldn't get it. And I thought that I'm not going to give them money. I may make all my tools publicly available. And so I think I should also get tools from other people free of charge, right? So, which doesn't happen. And they were asking for money. So I decided to change the project. I didn't do it. But you're right. Yeah, so there might be some tools. I think I can look for that. We can discuss at some point. Yeah, there might be. Okay, so for few minutes, I think you will do a tutorial with me. And this is the last tutorial. Number 4. And this is just we will discuss actually, and you can do later on. It's okay. So yeah, we'll do it quickly. Yeah, you will just try to answer the question, and we will go fast. So a sequence is given, and that sequence is TCA, TG, TGC. And for the HMM hidden Markov model structure shown above, to describe this sequence with hidden paths containing state E, E is exon, 5 is 5' splice site, I is intron. Okay. How many distinct hidden paths with joint probability not equal to 0, where S denotes hidden path that underlie observation sequence O? So how many paths have the joint probability not equal to 0? Okay, when you compute the joint probability of observation sequence and hidden sequence, that should not be equal to 0. So how many such hidden paths are there? Okay. So look at the observation sequence. So when it would be 0, so look at the values of the output probabilities. So suppose I say that the boundary is this C. So will the probability be 0 or non-zero? 0, because probability of C from 5 is 0, right? So okay, that would be 0. So how many will have non-zero probabilities? So I am looking at, okay, 1. So A also 5%. Okay. So 1, 2, 3. Okay. So there are 3 hidden paths with non-zero probability, and you know how to compute the joint probability of observation sequence and hidden sequence. We discussed. I'm sorry. How many? 2, not 2, 3, right? So A is 5% and G is 95%. So okay, so where A, G, G, 3. Okay. Okay. So 3 hidden paths with non-zero probability. Okay. So now the next question is, what is the most likely hidden path that underlies observation sequence O given the above HMM? So how will you compute this? You have to find the most likely hidden sequence that underlies this observation sequence, and once you have found the most likely hidden sequence, then you know where is the splice site, right? So how you will do it? Yeah, probability 95%, but still I don't know actually which is the splice side. So there are three paths. One is, okay, 5 is here. So 5 is here. This is the boundary, and to the left of the boundary, these two are exonic, and to the right are intronic, right? Then you have the third one, sorry, the second one, this G as the boundary, to the left exonic, and to the right intronic. Okay. So you have three paths. Do you remember how to compute the joint probability of the observation sequence and hidden sequence? Probability of starting in S1, then emitting O1, and then making transition to S2, then emitting O2, then making a transition to S3, then emitting O3, right? So that is the joint, that is the joint probability. Okay. So you would compute the joint probability for each of these three hidden paths, and then select the hidden path that is the highest value of the probability. Clear? So once you have determined the most likely hidden path, you know where is the boundary. For example, if the probability of this one actually, so I say that, okay, this is the boundary, so the third hidden path. So this is the boundary. To the left of this are exonic nucleotides. To the right of this is the single one, intronic nucleotide. Okay. If this hidden path has the highest probability value, then you know that G, this G is the boundary. This G is the 5' splice site. Okay. Okay. So that done, first question. Second question, choose a transmembrane protein and use the FASTA sequence as input sequence to FOROMICS program and TMHMM program. Okay. So what you have to do, you have to get a transmembrane protein from NCBI. Okay. So you can do it. So go to NCBI. Yeah, this you can do quickly. So I am at NCBI, so let's go back actually. Okay, and then I will look for protein. So protein. Protein and write transmembrane. Transmembrane. Okay, so terms of transmembrane protein, and you can select a long one, something, any you can select, it's okay. I'm just looking for a long one, so this one. So you can, I think it should accept the accession number, otherwise you can copy paste the FASTA. Okay, so FASTA sequence is here, and next you will get the TMM, HMM or FOROMICS. So TMHMM is for predicting the transmembrane topology. So click on TMHMM. So here it is, okay, and then you can copy paste. So here is the sequence, and copy, and then paste, and then submit. Okay. So it will run, I think it should run fast. So similarly you can do for the phobia, so it generated. Okay. So so how many helices? How many helices are there? Transmembrane helix. How many? No. So okay, so look at, where are the first few nucleotides? sorry, residues, amino acids. So this is the probability. So you will look for the highest probability. Which has the highest probability? Outside, inside, or transmembrane? Outside. So all these amino acid residues, they are outside, right? And then when you come here, then it goes into membrane. So it's transmembrane, okay? And then from this, it goes inside. So few nucleotides only inside, okay? That we can't see here. So how many helices? How many transmembrane helix? One. So which is shown here? One. Okay. So and you can do with phobias and you can compare. So this was the tutorial. So you should be able to interpret the topology of a transmembrane protein. You want to know which portions of the transmembrane protein is outside of the cell, which residues are within the membrane and which are inside of the cell, right? So this program will help you do it, and you can try with the other program phobias also. Okay, so let's go to next question. So yeah, if you are interested, then try this at home. You will learn it. So yeah, it's a long question actually, and again, I'm not asking you to do it, but this is about some protein. So XALD is the most common inherited disease affecting peroxisomes, a subcellular organelle involved in lipid metabolism and other metabolic functions. This disease is caused by mutations in the ABCD1 gene on chromosome X, encoding ALD protein, ALDP. In humans, there are thought to be 4 ALDP related proteins. The accession numbers are given. Okay, 2 yeast ALDP like proteins have been identified. Accession numbers are given. These proteins are all part of a much larger family of ABC, which is ATP binding cassette transporters, including the cystic fibrosis transmembrane regulator CFTR and multi-drug resistant proteins MDR. Create a multiple sequence alignment of the human, mouse and yeast ALDP family of proteins. So I have provided you the accession numbers for human and yeast. Can you get accession numbers for mouse from NCBI for this protein? Okay. So once you will get, just get for maybe 3 proteins or something like this, not many. Once you have got the accession numbers, then create a multiple sequence alignment of the human, mouse and yeast ALDP family of proteins. Identify the presumed nucleotide binding site. This is the binding site. Look at whether this is conserved or not. Okay. So this is the nucleotide binding site. You would expect this to be conserved. So you would examine the multiple alignment for this motif. Okay. Is this motif perfectly conserved? That is the question. Okay, or not? Perform multiple sequence alignments using programs available at European Bioinformatics Institute, and they are Clustal W or Clustal Omega, MAP, Muscle and T-Coffee. Okay. Save and compare each result. How do they differ? In particular, look for multiple aligned regions with GP and GC, GKS motif, and provide only a part of the alignment that contains this motif. Okay, you can do it. So you have to get comfortable with using these tools and understand what these tools can do. That way you will appreciate the usefulness of these tools, and that is why these questions are there for. Okay, not a big deal. You have learned how to run BLAST. You can do it. It's even easier than BLAST, multiple alignment. Okay, next question. So I'm about to finish. Then Dr. Sukla will come. Okay, use the sequences from the ALDP family of proteins as in question number 3 and construct a phylogenetic tree using a publicly available software tool such as MEGA. But for MEGA, I think it's a very large file I downloaded, and it's really very large. You can make a tree using cluster W itself. Cluster W provides an option to make a tree, and that's never joining tree or cluster omega. So click on a phylogenetic tree, it will generate you the tree. Okay, you can get the multiple alignment, then you will get the option to make a tree. Click on that, it will give you the never joining tree. Okay, so next question. Okay, so search for PFAM matches to this protein. So what PFAM does? Do you remember PFAM? PFAM is based on hidden Markov model of protein families. So given a protein sequence, what PFAM will do? Search for the conserved domain or the family that this protein belongs to, right? It will find you the family that this sequence of your interest belongs to. Okay, so you will go to PFAM and then just copy paste this one. So this is the last question, and go to, I think Dr. Sukla might be coming, so I have to do fast. Okay, so go here and sequence search. Okay, and copy paste the sequence. Paste and go. Okay, and it is now hosted by Interpro. So Interpro will do the work for you, and I think it is running. So it is searching actually, the top one. So it is running. So it will provide you the information regarding which protein family this belongs to, and also the domains in this protein. So maybe one domain or more than one domain. Okay. So yeah, it takes a while. Gaurav, you can text Dr. Sukla to. Oh, done. Oh, so he should be coming. Okay, good. Okay, so it is still searching. So, and the other question actually for RNA-seq, there is another tutorial which is number 5, and that's just one question, and this is take home. So, visit NCBI GEO and download an RNA-seq dataset of a small size, preferably microbial, that can be used for differential expression analysis. Describe the samples downloaded, control versus condition, and the problem being addressed. Okay. Use the Galaxy RNA-seq pipeline for trimming, quality check, alignment, abundance quantification, and differential expression analysis. List the differentially expressed genes and interpret their significance in the context of research question being addressed. Okay. So I would encourage you to try the RNA-seq pipeline at Galaxy. Okay, try it. I have provided you the screenshots. Why not try it with the accession numbers also that I think Sravani will forward to you all actually. The 4 access, yeah, do not use, to begin with, do not use 3 replicates, just use 2 replicates. Okay, try it and see what you are getting. Once you become comfortable with, then you can do with more replicates, and you can analyze the samples of your interest. Okay, but try it because you would want to have a feel of how this pipeline works, and it is important because this is one of the most used omics pipelines. The differential expression pipeline. This is not just Galaxy, but there are many other pipelines. This is very user friendly. It's a web server. So you have web interface you can do on your computer. Otherwise, you will have to download the tools and then install maybe Unix computer it will need, or it will be really too much of work, right? Which one?
There are many softwares that you can use for the same thing.
Yeah.
Differentially.
Yeah.
So which one would be the most accurate or the best one?
No, yes, use this one. Yeah, it's okay. HiSat is good for alignment. Sometimes people use a star, Salmon, and but then you may have to run on a Unix computer. This provides option of DSeq2, which is the most used tool for differential expression analysis. So just try Galaxy. I think it's a good one-stop place.
So in case we have our own data, like here we are searching the materials data.
Yes.
Versus computing, we have our
Own data, then just upload it.
on the same file.
Right. Yeah. You can just upload the Galaxies, right? Yeah. Upload there. So yeah, it did actually, and we can see actually which family it belongs to. Okay. So I will click on this one. Okay. Which family it belongs to? Answer question. Answer my question. Which family this sequence belongs to? Which one? UDP glycosyl transferase. So UGT family. Okay, and this protein is very important because this protein can detoxify our body. So it's so important. And for example, chemotherapy actually that destroys the body. It is for killing cancer cells, but it is so toxic. So people are doing research on UGT so that UGT can be used for detoxifying the drugs used in chemotherapy. Okay, so this belongs to this family, UGT family, and then okay, and it is asking for. No, yeah, come, आ जाइए, हो गया मेरा. Okay, हो गया. Yes, that's fine. Yeah, I'm done. Come, okay. Get started. So, can you all? I got you started. So, can you work on this problem? So, you have to also identify conserved domain. Okay, conserved site within this protein, and also get the conserved sequence as well as the sequence logo of the conserved site. Sequence logo, have you seen? You have seen, okay. So it will provide, okay. Okay, so
Okay.
yeah, set up.
Link on है ना? So we will have a quick recap.
Oh my.
रुक जाओ एक मिनट. Okay. वो कहाँ है इसमें? चलिए. If you want a break of 5 minutes, you can take. You want break? Okay. So you can take a break of 5 minutes. Okay.
Okay, break of 5 minutes. Okay, take a break of 5 minutes.
हाँ. किसका? हाँ, टेस्ट स्कोर अभी आएगा आपके पास. अभी चल रहा है मतलब 80% हुआ है, अभी 20% बाकी. लोडिंग है, लोडिंग. So 5 minutes break, then we'll start. लिंक आपने पॉज किया? अभी पॉज किया है? ऑन है. 5 मिनट का ब्रेक है. ठीक है.
हम सोच रहे हैं कि फिजिक्स डिपार्टमेंट थोड़ा देखते हैं.
अभी आपको ले चलते हैं. बस इसको फिनिश करिए. मेरा है, मेरा.
ये भी लग रहा था क्या?
हाँ. वो यशु को बुला लो. लिंक थोड़ा वो कर दो, या तो पॉज कर. तुमको आता है? हाँ हाँ अभी जब वो चेक हो जाए पूरा तो सर को साइन कराना है। तो अभी सर को लैब में ले जाके वहाँ मेरे उस पर बिठा दो, ठीक है? तो वो सब साइन कर देंगे, टीका वहाँ बैठी हुई है, ठीक है? फिर मैं आता हूँ इसको फिनिश कर। फोन करके पूछ